Center for Bioinformation. And let's welcome our first speaker, Jingweng Fang. Okay.
Hello? Oh, can you hear me? Okay. Well, I'm so appreciated to be invited to give a talk in such a great event. I'm Jingweng Fang from the University of Science and Technology of China. Thanks Easter to change my order since I have to catch my airplane. Today, I will share my recent preliminary work, scPERT, a single-cell perturbation-enhanced representation transformer, which we built by fine-tuning the scGPT on the whole 100 million single-cell data. In simple terms, scPERT predicts how individual cells respond to perturbations like drug treatment. Okay, so in the next few minutes, I will cover the following points. First, a brief background introduction about single-cell foundation model, supervised fine-tuning, and perturbation. And second, some details about scPERT consist of data curation, methodology about the novel LoRa fine-tuning strategy we devised, and the performance evaluation. Recent advance in single-cell sequencing have generated massive multi-model datasets, just like you hear in this HCA meeting. Foundation model, which means the deep neural networks pre-trained on large data, are now being applied to biology. For example, the scGPT was trained on over 33 million single-cell transcriptomes and have shown strong performance on many downstream biology analysis tasks like cell type annotation and batch integration. Based on these foundation models, a breakthrough biological concept of virtual cells now appears attainable. Over the past years, a wealth of paper has stretched the architectures and objectives of virtual cells, the foremost of which is to predict how cells respond and adapt under varying conditions. So our work, likewise, aims to take a step forward to the construction of virtual cells. Okay, so, sorry. Oh, there's some mistakes. Okay. So why we focus on post-train? Here, let's get a brief overview of the foundation model construction process, which generally splits into pre-train and post-train. Take DeepSeek, which is one of this year's most talked about models as an example. Its pre-training phase consumes 95% of the GPU hours, make it a resource-dominant effort. By contrast, the post-training phase used only 4% of the GPU time, yet encompassed the most critical algorithm and data. Indeed, DeepSeek R1 has not publicly disclosed its post-training GPU code and its 600 high-fidelity data for post-train. So that's why we focus on post-train, and on the other hand, we cannot afford so much GPU, in fact. So in terms of perturbation prediction, it plays a pivotal role in virtual cell research by enabling in silico simulation of how individual cells respond to diverse treatments or environmental changes. This capability accelerates the hypothesis generation, get experimental design, and helps identify candidate drug targets or resistance mechanism. Well, previous models have also attempted to fine-tune a perturbation prediction taskset. However, because earlier perturbation database was small and compiled from diverse experimental protocols, like shown in here, their data suffered from substantial structural heterogeneity, resulting in limited performance. So yeah, and in March of this year, the new giga-scale, the whole 100 million dataset were released, which originates from a single experimental pipeline covering 379 drugs across 50 tumor cell lines. The public availability of the whole 100 million has greatly enhanced the feasibility of perturbation prediction models. However, so little is what we do. So however, the whole data size, since the whole data size is it's produced by the drug treatment. However, it is well known that most small molecular drugs do not interact with genes in a strict one-to-one pattern. Therefore, our primary objective is to create rigorous drug-gene association and to filter for high-quality data. Firstly, drug information is extracted from drug bank and ensemble API, and additionally ascribed from the MedChem Express, okay, this website, is MedChem Express website using Selenium. The collected data is then summarized with the DeepSeek API to identify the output potential drug targets. Secondly, based on the aggregate drug target information from the multiple sources in the first step, an expert manual creation process is performed. The reliability of each entry is scored according to the consistency of source and the clarity of the mechanistic descriptions. Using the drug reliability scores obtained in the second step, we perform data cleaning based on QC and the confidence score of the drug and the relative gene. And furthermore, we develop a new LoRa, which means low-rank adaptation model. This part of work was carried out by my collaborators at the Harbin Institute of Technology. In briefly, our post-train model consists of non-parameter path selection, dynamic path adaptive update, and orthological constraints, which leads encouraging each adapter to learn different aspects of the data, encourage diverse representation, and improve the adaptation capability without increasing parameter count. Now here's our performance. Let me see. Okay, based on the bowel approach, we trained our model on the high-quality subset of the whole, which means it's 10 million, and evaluate using both few-shot and zero-shot data sets. In the few-shot data sets, the model encounters the same cell types as in the training site, but to predict the drugs, predict the different drugs. The UMAP plots of the predict gene expression shows that our model, which is the LoRa tuned and the 30%, will outperform the other model like the SGPT base or full parameter fine-tune and the other LoRa method. Likewise, on the zero-shot data set, which is completely unseen data from scPERT, it seems also achieved superior performance. Furthermore, statistical metrics such as MSE, RMSE, and L2 loss and consent similarity also demonstrate that our fine-tuning approach outperforms on both the base model and the conventional LoRa models. Furthermore, we integrate transformer and convolutional neural network architecture for reverse gene perturbation detection, which is built up on a modified ResNet 101. This module transforms four-dimension gene features, raw expression, predict expression, their difference, and absolute difference, into a 512 plus 512 feature maps. Deep features extract via the enhanced ResNet 101 and pass through a multi-layer perceptron to output gene-level perturbation probabilities. On the amazing dataset containing 5,000 genes, the model achieved the top K hit rate of the best of the 11% to the 27% for the inverse prediction. Okay, so thanks for my teammates, thanks to my professor, Professor Kun Xu in USTC and my lab members, and also thanks to Professor Fan Xiaopeng, Wan Zhaolin, and their lab members in HIT, and also Zhejiang University, Jia Xuan and Yu Yang. Thank you for attention. Leads us their preliminary results. We have many benchmarks and downstream works in progress. We greatly welcome your feedback and suggestions and invite my research interested in AIVC development and contact me at any time. Thank you.
We don't have time for questions.
Okay.
Okay, so our second speaker is Herawati Sudoyo. Yeah, let's welcome her.
Good afternoon, everyone. Let me share with you our experience in Indonesia regarding the mapping of immune cell diversity across Indonesia, integrating the genetic, lifestyle, and collaborative insight. I think this slide has been used for 3 days, meaning that this is important and this is our concern. This slide has been used widely, actually, every time that we have to present our activity. We know that we are underrepresented and we are ethnically diverse population. That underrepresentation will impact our ability, actually, to fully understand the disease and, of course, the genetic architecture of the and also exacerbated health inequities. Why Indonesia? I am not going to tell about island of South Asia, but Indonesia. Indonesia is a dynamic theater of human development and migration. We are a maritime continent, we are a maritime country with almost 16,000 islands, straddling the equator approximately 3,000 miles. It is home to around 275 million with almost 500 diverse ethnic population speaking 719 languages. So we found that we have a complex pattern of admixture across the Indonesian archipelago by the study from Hugo, Pan-Asia SNP Initiative, Genome Asia 1000K, and 10,000K, and others, including IGDP. We know that 60 to 50,000 years ago, there is a settlement of present-day Papua New PNG and Australia, and we found Denisovan integration in the East Asia. 5,000 years ago, of course, expansion of the Austronesian speaking language farmers into Western island of Southeast Asia. So now what we have is geographically structured genetic Asian cline. We have been working with the Indonesian diverse population from Aceh in North Sumatra in the west to Kirom in the Papua, in the border of West Papua and Papua New Guinea. Up till now, we have studied more than 130 ethnic populations, collected more than 5,000 samples with 3,000 human whole genome sequences data and more than 500 genome-wide data. What we study is not only genetics, but we also collected words of different languages in Indonesia. We also studied the culture and local wisdom, and of course, clinical information. We cannot work alone in this research activity. This study is supported by CGI and a strong collaboration with St. Vincent Institute, TGen USA, Massey University of New Zealand, and of course, the most important thing is the local university. The project will cover, the innate immune system. So we look also at genetic ancestry and looking for ethnic or population specificity. The adaptive immune system, we look at the role of infectious disease exposure, environmental and lifestyle changes. And of course, study the cellular state differences through gene expression and gene regulatory interaction. We chose to study population, not all of the Indonesian, due to the lack of funding, of course. First, the Balinese. Balinese, which is now in transition from agriculture to tourism, from traditional life to urban livelihoods. 40 years ago, the economy based largely on agriculture. Today, tourism is the largest single industry in Bali. The second one, what we choose, is from the eastern part of Indonesia. We look at the dynamic geography of Papua, contrasting highland and lowland. So, what are we doing at the field? This is not a simple study. This is not a simple activity. This is a very complex activity. The challenge that we face working in country with very diverse ethnic, language, and tradition is that one size does not fit all. We have to adjust accordingly to what we face in the field. So we have to do, I can't see from here, working with the local authorities. We have to do some sampling site determination, addressing local logistic issues, involving the local community, which is important. Then the last one, doing the sample and data collection. First of all, there are a lot of processes that we have to do, permits from local authorities, universities, local laboratory and healthcare involvement, and the most important and the key for success of this kind of activity is the local community itself. It's not only permit, but we also have a community engagement, a very strong one. The other one is, of course, this is all of the process that we have to do. We set up kind of place that we could do and we could inform the local community, registration, inform consent form, all of the blood pressure, blood collection, and health and diet interview, which will take some time, and many, many tests. So capturing Indonesian lifestyle diversity for this project, we chose Bali, 112 donors, 60 from urban and 52 from rural. From Papua, we have 91 donors, 43 from highland, and 48 of Yoboy. Why we choose that? Because we have a data on the admixture. So we choose the one which is representing the west part of Indonesia, which is Austronesian-speaking population, and the eastern part of Indonesia, Papua, Papuan Lak with Papuan-speaking population. We have challenge, and that's actually something that we have to do and to check whether they will give us a meaning of a result. Challenges in sample transport. When we actually did the collection in Bali, we have to do 4 transportation to the local laboratory from the university. And it took about 0 to 24 hours from collection before it was actually isolated, before PBNC is isolated. And also from Jayapura, it will, because the distance is very long, you can see here, that sometimes you have to do stop over here. Will take about 6 hours to go to Jakarta to our own lab, because no one can do the PBMC isolation in Papua. And that will take about 12 to 40 hours from collection. So what we did with all of those RNA sequence and VDJ multi-omics only for 8 samples per site, and for all samples we do shallow pass. So with this, annotated the samples. This is what our UMAPs look like. We have captured over 480 to 565 cells across 199 samples, and for now have done 2 levels of manual annotation. The challenge now is to carefully analyze our data to take into account technical batches so that we can focus on meaningful biological insight. We have adjusted for age, sex, and sequencing batch and duration when looking at differential cell type abundance, and found significant differences between the comparison. 8 cell types show significant differences between ancestry, Bali versus Papua, which is here, and then also a donor site representing environmental variation, show differences in 2 cell types within Bali and Papua, highland versus lowland. This is also adjustment for biological and unwanted technical variation. We also show significant cell type specific differences between ancestry group. Individuals from Bali have greater effector memory of CD8 on the left side than Papua, and on the right side we can see that the Balinese have a less expanded memory as compared to the Papuan. We can look at this study was done by Pongsakorn, Sukotaman. This is the slide that's showing differential gene expression and functional analysis of expanded clones between ancestry. The site shown that the clone exhibit activity in pathways related to immune cell activation, migration, and addition across both countries. The sign showing the regulation of viral processes and negative regulation of viral processes, which is enriched in Papuan. So what next after this kind of things? We expand our collaboration, which allow next step and capacity building or capacity development with Luis Barrero of University of Chicago. The capacity development is actually involved 4-week hands-on training for our member at University of Chicago. He had gene expression profiling and chromatin accessibility with scale biosciences here. There are 32 samples were stimulated with lipopolysaccharide and influenza A virus. And we are now still waiting for the progress for sequencing result. More result to come and more collaboration to come. Collaboration, collaboration, and collaboration. We have two kinds of projects, which is, and the Indonesian Genome Diversity Project was supported by many, many collaborators. That's the aim was actually to study genomic and functional genomics of Indonesia population and its association with susceptibility to complex and infectious disease, genetic variation and microbiomes of non-communicable disease related to lifestyle. And the third one, immune cell atlas of environmental and ancestry diversity in Indonesia. That's the single cell project. We collaborate with St. Vincent Institute, TGen, as I mentioned, Massey University of New Zealand, and we are still waiting for others. I think with this, I would like to thank you all for your attention, and we are looking for another collaboration. Thank you very much.
Let's thank Herawati. Welcome our next speaker, Hideki Ueno.
Good morning, everybody. And thank you for inviting me to give a talk. And thank you for staying on the day 4 of the HCA meeting. I'm so excited. So I'm Hideki Ueno from Kyoto University. And so I'm going to talk about the human liver immunology. So very brief introductions, because I'm really new to HCA. So I graduated Kyoto University as MD, and I did the pediatrics department for 9 years. Then moved to basically US and stayed in the Baylor Research Institute for 15 years. And I became PI, and again moved to Mount Sinai 2016 as a professor. And then I thought I would stay forever in the US, but somehow I got the offer from Kyoto University and went back to Kyoto University. Particularly after the pandemic in 2020, I needed to choose either Kyoto or Mount Sinai, and I chose to come back to Kyoto University. So what my point in this slide is really I'm a human immunologist, dealing with human samples, doing immunology for more than 25 years. So the strong belief is that there's a huge difference between the immune system, between mouse and humans. And so that's why we're really all the time dealing with human materials. So far, we have dealing with many times in human blood samples, because that's really easy to ask to clinicians, and it's really accessible. But then technology-wise, it's really developed in the past decades. Now we thought maybe we can do something with other than the blood samples. So then when I went back to Kyoto University, I wanted to do something with human organs, immune cells, because immune cells are not only circulating in the blood circulations. So immune cells are all, many actually immune cells are residing in the different organs, including many tissues like brain, lung, and the liver, and the colons, and everywhere. So I needed to choose some organs that I wanted to focus on in Kyoto University, and my target was the liver. And to really understand the liver diseases, so we need to deal with not only blood samples, we need to get the liver samples. That was really my strong belief. And then I think we are very lucky in Kyoto University, because there are many liver transplantation cases in Kyoto. So basically, when they do the liver transplantations, they took some, this way, donor liver, and part of this donor liver, Before this go to the recipient, they basically wash out all the immune cells inside the liver. It's so-called perfusion. So basically put a pipe into the portal vein and the hepatic vein and pushing out all the immune cells to the out of these hepatic arteries. So these immune cells localized in the human liver are all washed out inside the liver perfusate. But for the liver surgeons, they don't care about this perfusate. They're just unnecessary ones. So basically, as an immunologist, we are taking all these cells for the immune research. So as you see here, what we are collecting is basically they are putting a liquid from here to here and getting out of the liquid here, meaning that we can get all the immune cells residing here, this so-called sinusoid. And actually, many liver immune cells are residing in this sinusoid. So basically, that means that we can get most 100% of the immune cells in the liver by dealing with a liver perfusate. And not only a donor liver perfusate, so we are also dealing with the excised diseased liver. So this is a kind of many kind of different reasons. So they need to change the livers, and we are getting out of this excised liver. Then we do liver perfusions. So my students go into the pathology department and putting lots of liquids into the portal vein and washing, flushing out all the immune cells. We are collecting all of these samples. So as you see, this perfusions works very, very well. So this is the case of the hepatoblastoma. So these black dots are all the hepatoblastic tumor cells. But as you see, the rest of the liver is beautifully washed out, meaning that the liver immune cells are really washed out during these perfusions. So what we're doing is that, of course, we want to know, what the immune cells in the human liver, and also we are kind of dealing with many different kinds of immune diseases. The many immune diseases can be caused by the kind of dysregulation in the human liver, including excessive immune response. It's like autoimmune hepatitis, primary biliary cholangitis, primary sclerosing cholangitis, and biliary atresia. And also, two-week immune response can cause a disease in liver, including chronic hepatitis B and hepatocellular carcinoma. So basically, we are collecting all these disease tissue, liver tissues, and get liver tissues as well as liver perfusate to basically to collect different kinds of the immune cells in the human liver. So as I said, so this is what we're collecting. We have been collecting for 4 years now, since 2021. And we're collecting the human normal donor liver cells, liver perfusions, and the liver tissues, and diseased liver, liver suspensions, and also perfusate. Also, we're collecting liver biopsies. Like after transplantation, they do a liver needle biopsy in the multiple times. So we're collecting all these tissues as well. It has a really fundamental resources to understand human liver immunology. So this is what we have collected so far since 2021. So donor liver perfusate, we have collected more than 200 cases, and including 20 to 65 years old. That still range very wide. So we can get very young liver from old donor liver. And the male to female ratio, we made it 1:1. And we have collected more than 100 cases of the liver disease livers, PBC, PSCB, and SCCs. So that's created lots of liquid nitrogen tank. So we need to pay a lot for liquid nitrogen now, but now it's kind of fun part. So what we are doing is immune system in the human liver is very complex, but very little is known actually, particularly in the humans. So that contains many like different types of macrophages, T cells, B cells, NK cells, ILC, so on. So basically, we can collect all of these cells by the perfusate. At the beginning, so our concern was maybe by collecting the perfusate, then by containing lots of blood contaminations. But as you see, it's very simple. The FACS analysis comparison between peripheral blood and the liver perfusate. As you see by looking at, in this case, we're looking at the T cell compartment. They're hugely different. And these cells, like tissue-resident memory T cells and the main cells, only reside in the liver, not in the peripheral blood. As you see, we really have fundamental enrichment of the liver residing immune cells in the liver perfusate. So by taking liver perfusate, by using simple FACS analysis, as you see, we notice that we contain many variable immune cells in the human liver. And many contain like tissue resident cells, like a tissue resident CD4 T cells, CD T cells, gamma delta T cells, and also even NK cells are really localized, many in the liver tissues. So what we're doing after we're collecting, so we'll keep on collecting these samples. What we do is basically we do multiple single cell analysis, including multicolor flow cytometry, and also of course we do many single cell RNA sequencing plus CytSeq, and with also spatial gene analysis. All this equipment are already available in Kyoto University, so we can do basically everything. So this is kind of really easy comparisons on the cirrhotic liver versus normal liver perfusate, looking at the composition of the immune cells. As you see, there's really many differences, like lymphocytes lower in the cirrhotic liver, and the increase of the granulocytes, macrophages, so on. So that's really easy kind of difference, easy kind of examinations. And also we do many single cell RNA sequencing, so that's really many of my students are involved for this project. So I want to show kind of really preliminary results, showing kind of the difference of the immune cells between liver and the blood. So here I believe we did two liver perfusate derived single cell RNA sequencing, and we can see kind of many different kinds of immune cells. But one cells we focus on in this study was CD4 T cells. The Murakami-san presented yesterday talking about his new technology to identify the basically enhancer sequence by using 5' single cell RNA sequencing data. So we collaborated with him basically to look at what kind of new enhancers can be found in a human liver CD4 T cells. And we tested his database and containing that maybe many cells, blood CD4 T cells, and he identified more than 60,000 new enhancer sequence in blood CD4 T cells. But as you see, in a very small number of CD4 T cells in the human liver, we already identified more than 400 new enhancers only found in the liver perfusate, in the liver CD4 T cells. It's really shows that there's a very fundamental biological differences between the human liver CD4 T cells and the blood circulating CD4 T cells. So most important part of this presentation is that here. So now we are kind of redoing a single cell RNA sequence on 72 perfusate samples, and including 48 normal donors, age from 20 to 65, male to female ratio 1:1, including 24 diseased liver. And we are kind of expecting to finalize this sequence in this year. So in that way, we can really compare the kind of aging, the impact of the aging of the immune cells, including like Kupffer cells and NK cells, T cells, and to see how they changed by the age in the liver microenvironment. And of course, we can get some insights from human liver diseases as well. So I want to show a little bit, maybe spend maybe 2-3 minutes talking about a little bit subset of CD4 T cell we are kind of focusing on in this project. So as you see here is a CD4 T cell in a liver perfusate, and it's a FACS analysis, and compared to the blood circulations, as you see there's a really big area that can be found in the liver perfusate, which are of course tissue resident CD4 T cells, which express many markers associated with tissue residency, including CD69, 606, and the CD49A, and so on. And we do many single cell RNA sequencing to see what's really unique to this liver CD4 T cells. And then, so this is a kind of some kind of analysis of the CD4 T cells isolated from the 5 liver perfusate. As you see, that contain many different subsets within liver CD4 T cells. And many are kind of unique to only liver perfusate. And also we focused on one subset, which is tissue resident memory CD4 T cells, which is very little known even in the world about the nature and the feature of the biology of the cells. So by getting to CytSeq, getting to CD69 positive T cells, we wanted to see how heterogeneous they can be. And as you see, by getting to 69 positive, so-called classical tissue resident memory CD4 T cells, as you can see, the really heterogeneous populations, including many different subsets, including like a CD4 CTLs expressing granzyme B, granzyme K, and the Tregs, very minor population, but they do exist as a tissue resident regulatory T cells, and many Th1 cells, and some Th17 cells, and some stem-like cells. So as you see in a liver microenvironment, CD4 T cells are residing as a kind of different subset, and we are looking at the kind of impact of aging to these subsets and how the diseases affect these subsets. So the conclusion, there are still many unknowns in the immunology of human livers, and we want to really go into depth in collaboration with the Human Cell Atlas and the liver, particularly liver groups, and also immunology groups as well, of course. And the multi-platform single-cell analysis approach will significantly increase our knowledge in local immune responses of human tissues, and establishing strong domestic as well as international collaborations will be the key to find new ways to fight against liver diseases. So I stop here, and thank you to all my lab members. I have a very big lab, so that grows like during the past 4 years. And many students, and half of the team are really focusing on the human liver immunology. And I want to thank Murakawa-san for the analysis of the retake, and the pathology department, as well as the hepatobiliary transfusion department. I stop here. Happy to take any questions. Thank you. Doesn't work. Doesn't work. Doesn't work.
Thank you. Really impressive work. Really amazing. I was just wondering, in your cirrhotic population, what is the underlying chronic liver disease that you have in your cohort, and any intention to move towards MASLD patients as well?
Yeah, so we have many kind of patients because we are kind of how many 10 cases per month liver transplantations. So all of them basically have cirrhotic livers. So we are not really actively collecting alcoholic liver or NASH or MASH patients, but I think we can collect any time. So for the moment, we are focused on 4 diseases as I demonstrated: AIH, PBC, PSC, and BA.
Okay, thank you.
And HCC as well.
Yeah. Hello, I'm from India. So impressive work, sir. My question is, when you collected the transfusate and do the immune profiling, was there an effect of treatment? Because it must be from different causality that the patients went through transplantation.
Okay, so for the moment, so now our purpose to make a human liver immune atlas now. So by using by focus on donor livers, of course there's no treatment to those donors. For the recipient for the disease liver, of course there's kind of differences in kind of treatments in each patient. So we can getting all kind of data from together with the liver tissues.
Okay, thank you. Wonderful work, sir. I would like to talk to you after the meeting.
Thank you.
Thank you so much.
All right, thank you.
Thank you. Let's move to next speaker, Christine Zhang.
Hello, my name is Christine, and I'm from Seoul National University in Department of Biomedical Sciences. First of all, thank you so much for giving me the opportunity to present my work today. I'd like to introduce you to my study on elucidating gut immune signatures in Korean inflammatory bowel disease cohorts. So I have no conflicts of interest to disclose. So inflammatory bowel disease, or IBD, is a chronic relapsing remitting inflammatory disorder that affects the gastrointestinal tract, primarily consists of Crohn's disease and ulcerative colitis. IBD has predominantly been observed in Western populations. However, we've recently seen a rapid increase across Asia, especially in South Korea, likely driven by industrialization, dietary shifts, and environmental changes. And despite this growing burden, immune atlases and reference data sets focusing on Asian populations are significantly underrepresented. So therefore, there's a critical need to develop population-specific immune atlases that capture the distinct immunological features of East Asian patients. So today, I'm very excited to share the initial steps of our project, building a comprehensive single-cell atlas of gut tissues from Korean IBD patients. So our primary goal is to uncover the shared and distinct immunopathogenic mechanisms that drive UC and CD within the Korean population. And also, we're looking to identify immune dysfunction that are specific to our cohort. And looking ahead, we also have plans to perform comparative analyses with non-Asian IBD cohorts, and this will allow us to pinpoint population-specific immune signatures, ultimately paving the way for more tailored and effective therapeutic strategies for Korean IBD patients. So there are great public IBD single-cell data sets available, and they've given us a ton of information. They've helped us find new cell types, understand complex immune interactions in IBD. So while we're definitely building upon all this amazing existing knowledge, we're now creating a comprehensive single-cell atlas specifically for Korean IBD patients using our in-house data. So to start, we've focused on collecting both inflamed and non-inflamed gut biopsy tissues from the small bowel, ascending colon, and rectum of adult IBD patients. And we received these samples from Professor Sung-Jun Ko at Seoul National University Hospital. And because the hospital is so close, we can begin processing them within just 30 minutes of collection. For single-cell dissociation, we use a combination of mechanical and enzymatic digestion. We also fix tissues to create FFPE blocks, which we'll use for spatial transcriptomic analyses. We also collect and store paired PBMC samples from these patients, and to date, we have collected and archived samples from a total of 220 adult IBD patients. For single-cell RNA sequencing data generation, we thaw our archived samples all at once and multiplex them into groups of 4. We then use SNP-based demultiplexing with paired PBMC samples. So far, we've generated single-cell RNA-seq data from 152 adult IBD tissue samples for about 81 patients, and this includes 106 samples from UC patients and 46 from CD patients. And importantly for our UC data, we have both pre- and post-biologic samples as well. And we've recently generated data from healthy gut tissues to use as a comparison for our analysis. So for today's talk, I'd like to briefly introduce you to our gut tissue data set. As we're in the early stages of our analysis, we began by comparing the major cell subtypes found in adult Korean IBD patients. And looking from left to right, you'll see data from healthy controls, then UC non-inflamed and inflamed tissues, and CD non-inflamed and inflamed tissues. So it's already clear that there are distinct differences in cell frequencies across the conditions, and we're actively working to understand the biological implications behind these variations. Given our specific interest in the gut's immune cells, we started by dissecting T and KT cell subsets within these tissue samples. Out of all the cell types we've identified, I wanted to highlight the ones that show the most significant differences and reveal the disease-specific finding. So our group is particularly interested in regulatory T cell subsets in various inflammatory diseases. And as you can see, aligning with observations from Western IBD datasets on the right, our Korean IBD dataset also shows a clear increase in Treg cell frequencies in both UC and CD when compared to healthy controls. Interesting observation was a disease-specific increase in the T17 population within our Korean patient cohort. This finding kind of led us to delve deeper into T17 subsets for further analysis. To understand the immune dysfunction in Korean IBD patients, I focused on the Treg and T17 cell populations, annotating them with specific functional markers. I then subsetted these into 5 different clusters to examine frequency differences across our patient groups. What's most interesting is that the TT17 population showed a highest proportion in CD non-inflamed tissues. While this might seem counterintuitive, as TC17 cells are generally known to be increased in active CD, this finding potentially suggests a different role for TC17s in Korean CD patients, perhaps indicating a protective role rather than a pathogenic role in this context. Beyond just frequencies, I've also compared the functional markers within Treg subset to uncover differences in gene expression between our groups. Looking at Tregs, we observed increased IL-17A and decreased FOXP3 expression in UC inflamed tissues. And this potentially suggests that Tregs in inflamed UC are more pro-inflammatory and perhaps even dysfunctional compared to other patient groups. Beyond T-cell subsets, we also investigated other immune cells, like myeloid cells, and we've already seen some important differences, significant differences across the groups, and we have plans to investigate further in the future. Similarly, for B cells, they play a key role in follicles through TB interactions, essential for germinal center reactions, so we'll be looking into the differences between these groups more thoroughly as well. Moving forward, our plans include significantly expanding our single-cell RNA-seq data generation using our archive samples of about approximately 220 adult IBD patients, including both tissue and PBMC samples. We also intend to integrate this adult IBD data with our in-house pediatric IBD data set. This pediatric cohort is particularly valuable as we've collected samples from 7 distinct gut regions per patient. And for these IBD patients, we've already generated PBMC single-cell RNA-seq data and also performed CyTOF for protein validation. Furthermore, to gain spatial insights, we plan to generate spatial transcriptomics and proteomics data using platforms like GeoMx, Xenium, and Maxima, all from TMA sites sourced from the same individuals in our existing single-cell RNA-seq cohort. I would like to thank my professor PI, Hyundai Kim, incredible physician scientist who provides outstanding support to our group, and a big thank you to our collaborators, as well as for the funding from SCADE, led by Professor Jong-il Kim. And with that, I'll take any questions. Thank you for listening.
Great. We have time for 2 quick questions.
Thank you. It's a very comprehensive presentation, very great. My question is, would the composition of the gut microbes influence also the profile? Did you also consider to the gut microbiome as a confounder?
Right. So we have a different grant from Korea involving IBD microbiome. So we have plans to integrate that into our data as well in the future. Thank you.
Great talk. Any correlation with PBMC markers to your IBD patients?
Right. So I'm still looking into the PBMCs. I haven't done it yet, so maybe there might be some interesting findings. I'll let you know later. Thank you.
Hi, great presentation. Do you also have any data related to diet intake of those?
Right. So also for that, we do have clinical information for that too. So maybe we can integrate that to our data set, and that'll be great. Thank you. Thank you.
Now we are starting to our second session. Is first speaker is Sowon Choi. I hope I pronounce it correctly. Oh, sorry. This is please. Yeah. You have 10 minutes.
Thank you. Thank you for a kind introduction, and hello everyone. I'm Sowon Choi from Seoul National University. Slide not prepared, but in South Korea. Thank you. Hello everyone. I'm Sowon Choi from Seoul National University in South Korea, and I'm studying in Dr. Hyunjae Kim's lab. I really appreciate the opportunity to present my work in this meeting. My main topic for today is high-resolution spatial transcriptomic profiles across multiple cancers in Korea. I'm sorry. In past years, global efforts have been made to establish Human Cancer Atlas. As a result, the Human Tumor Atlas Network is constructing three-dimensional atlases of the cellular, morphological, molecular, and spatial features of human cancers over time. By contributing our spatial data set, we believe we can provide not just validation of existing tumor atlas data, but also additional insight into the mechanism of pathogenesis and resistance. Utilizing the Xenium 5K platform, we can annotate specific cell clusters and explore their composition and interactions. These enable us to unravel tumor microenvironment like tertiary lymphoid structures related to prognosis in particular cancers. Moving on to our sample collection workflow, from 7 collaborators across 2 medical centers, we obtained tissue samples representing 4 cancer types: glioblastoma, triple-negative breast cancer, pancreatic ductal adenocarcinoma, and colorectal cancer. Importantly, we collected not only tumor regions, but also various normal and border regions. In order to profile many patients and tissue areas efficiently, we made tissue microarray with 1 or 2 millimeter size cores. The picture to the left is a representative image for a CLC slide. With these TMA slides, we produced genome data using 5K panel. In total, we collected 430 samples from 180 patients encompassing 3.8 million cells prior to quality control. Additionally, all samples are labeled with clinical data, such as treatment status, the ratio of TIL, and progression-free survival time. These data can support to find prognostic markers in spatial data. To introduce our data quality and analysis pipeline, with 5K panels, we achieved a median of 233 transcripts per cell across all samples in average. As you know, Xenium is probe-based technology, so we check the decoding quality of gene-specific transcript. We can see purple dots representing pre-designed genes are separate from red dots representing negative control probes. Overall, we believe this indicates successful generation of high-quality spatial data. Also, we applied cell morphology stain and onboard segmentation analysis. In next step, we loaded the data using scPERT and performed cell and gene filtering as these criteria. Then we integrate and correct batch effect using SEVI. In the analysis step, cell annotation was carried out manually, guided by cell typist predictions and expert pathologist input. Furthermore, in order to investigate spatial context and cell-cell interactions, we employed the SciMap packaging. Notably, we identified spatial domains organized around distinct cellular niches using SciMap. Following cell quality control, we retained a total of over 2 million cells. First, we looked into the ratio of TME to tumor cell population in individual cancer type. Whole 4 tumors have non-lesion area except GBM. Notably, PDAC exhibited the smallest proportion of tumor cells in tumor regions relative to the other cancer types. And GBM displayed the largest proportion of tumor cells in tumor regions, especially in the PAN, which means pseudopalisading necrosis region. Let's look at the UMaps of whole cell clusters. Orange areas indicate tumor cell clusters, while blue areas represent non-tumor cell clusters. As evident in the orange areas, tumor cell clusters show prominent heterogeneity. Let's briefly look into cell composition of TME clusters per region in each cancer type. CLC includes a lot of inflammatory myeloid cell clusters in tumor regions. PDAC displays similar cell composition across all regions. GBM has rarely T cells and many macrophagy subtypes enriched in necrotic region. Finally, TMBC shows increased T cell and B cell population in tumor regions, suggesting the potential presence of tertiary lymphoid structures. Using the algorithm based on clustering in the SciMap toolkit, we identified 12 distinct spatial domains within the tumor regions in TMBC. We see spatial domain 9 is expected to have lots of T cells and B cells, considering their gene expression in heat map. We believe that this may indicate potential tertiary lymphoid structures. When we compare the spatial domains between low TIL and high TIL groups in TMBC, they show thoroughly distinct spatial domain patterns. And we can see that high TIL group on the right contains spatial domain 9, which is marked by yellow dashed line. We checked the cell distribution and location on genome image again. We can see the high proportion and co-localization of T cells and B cells in high TIL group compared to low TIL group in the location of domain 9. The TMBC samples are all treatment naive in early stage of cancer. We plan to investigate how TIL density and TLS formation relate to patient prognosis in a follow-up study. Finally, I'll be finishing off by sharing our further plans. We are integrating the cell clusters across multiple cancers. As we integrate 3 types of cancers, we found common TME clusters. Then we will try to reveal shared or unique spatial interactions across multiple cancers. Also, we plan to improve cell annotation and phenotype definition by referring to the single-cell data sets. Also, we will produce more data from serial sections of slides using other modalities such as Maxima and GeoMax. This is our group and collaborators. I'd like to thank the all professors at Seoul National University Hospital and Assam Medical Center for diverse tumor FFP blocks, and also all the students contributing to data generation. This work was supported by the Korean ARPA-H program, and we hope it will help advance drug development for intractable cancers. Thank you for listening.
We have time for questions. Anybody want to ask?
Great to see integrative analysis across multiple cancers. I will zoom in on one though, the colon cancer. What was the clinical phenotype you were trying to understand?
We have the MSS and MSI high CLC patient. So we try to compare the macrophage phenotype and T cell phenotype between the 2 subtypes. Yeah, and we also focusing on the regions of CLC like invasive margin and the tumor centers.
Okay, great. Look forward to those results.
Thank you for question.
So I'm quite interested in your tissue microarray strategy to, of course, save costs, but at the same time, the field of view is much smaller, or the area that you to investigate. So what are your thoughts? Again, it's cost-benefit question is, do you think we are losing out on potential, are we being biased by having smaller region, because sometimes tumor can be much larger, versus the cost factor to profile all these patients? So Any thoughts on pros and cons of having tissue microarrays?
Yes, right, you are exactly. And so we try to need more duplicate or triplicate regions from one patient, and also we should compensate the deficiency of information from single cell RNA-seq data, and multiple modality of platforms. So we try to.
Great.
Thank you.
Thank you.
One more
Hi.
question?
Yeah, at the back. Thank you. Wonderful talk. 2 questions. One, do you have the genomic signatures or whole exome-seq of the tumors so you can correlate with the different cell populations that you find? And then second, following up on the question from Jay about, the field of views or the FOVs, did you include or are you including like tertiary lymphoid structures? Because that's also part of the tumor biology that we kind of lose out when we're only looking at the epithelium and the stroma.
Yes, thank you for the question. And first one, we have some clinical data about genomic alteration and variations for CLC and GBM. So we can compare the patients between alternative genomic variations. So, and then the second question, we should consider the whole structures from the whole tissues, which is origin blocks from the TMA cores. So we should try to compensate the deficiency. Is it enough?
I think it's just, yes, thank you, that's fine. But just in terms of like when we're designing these experiments,
Yep.
What are we putting on the slide? What are we asking in the context of the tumor cells, the stromal cells, the cells, the tertiary structure, the tertiary lymphoid structures, which I thought to also be correlated with response to treatment or response to immunotherapy. And presence or absence in the tissues. It's not necessarily something you have to answer. But thank you.
Thank you. Yeah.
Thank you. Big applause. And the next speaker is Reema Baskar. You have 10 minutes.
Thanks. Hello. Okay. Hey everyone, I'm Reema, and thank you for having me here. So this was the original talk of my title, but I've learned some lingo in the last few days, so I've revised it to now extracellular cartography of CRC. Awesome. So colorectal cancer tumors are made up of very many different cell types, and crosstalk and communication between them creates these immunosuppressive microenvironments that drive tumor progression. And studies have modeled this a fair bit with spatial transcriptomics. But this begs the question, right? What are these key communication factors that are acting between cell types in the TME? And more importantly, where are they? So in order to bridge this gap, we wanted to look beyond cells and their spatial distribution to the extracellular space in the TME. So this space is highly structured with secreted and ECM proteins that are involved in signaling between cells. So in order to study this comprehensively, we looked across tumor locations of about 250 patients with around 71 antibodies to key protein markers. We used the Mibiscope platform for spatial proteomics at around 0.4 micron resolution and have patient-matched single-cell and spatial RNA data, along with key clinical features, of course. So our antibody panel encompassed several key immune function markers, metabolism markers, intracellular signaling markers, as well as around 18 proteins that are extracellular. So the study had a few challenges that we needed to build experimental and computational approaches to tackle. We needed to scale pipelines to handle large amounts of imaging data. We were also working with archival FFPE tissue with varying tissue quality and epitope degradation. And most importantly, we needed to develop a new vernacular to robustly characterize the extracellular space of the TME. So the big question is, what's outside cells and how can we capture it? So we clustered extracellular pixels across our 18 proteins of interest, which was billions of data points, and then segmented contiguous pixel cluster to get these extracellular objects. Here's a UMAP of our extracellular objects by protein, and here are CRC images with object segmentation masks. So here we see a mucin protein, TFF3 in blue, and a serine protease, HTRA1 in magenta. In a serial section, we see these proteins spatially corresponding with interferon gamma in blue, as well as TGF-beta in magenta, as well as the segmentation masks next to them. So next, we fine-tuned a deep learning model to denoise the data and correct for signal bleed over, as well as go ahead and cluster it for cell types. We are able to get these pretty UMAPs. They're able to segregate cells by their lineage. So we get these epithelial cells, of course, expectedly, lymphocytes, myeloid cells, and stromal cells, which really let us characterize FOVs at a pretty high resolution. Here we're able to detect invasive CRC, tertiary lymphoid aggregates, macrophage-infiltrated regions, as well as these crypt structures. And again, we're able to pick up extracellular things like TFF3, which is a mucin protein and secreted into the crypts. All right, now that we had done all this work, we wanted to meaningfully link these extracellular protein depositions to cell types and states. So we were interested in the patterning around cells, as that was most likely to affect cell state and modulate cell-cell communication. So essentially, we wanted to see what each cell sees in the TME. So we fit these concentric circles around each cell and traced out average protein abundance from cell boundary to about 40 microns out. And we took these millions of cells by 18 proteins by 100 pixel matrix and condensed it down into a weighted average per protein per cell. We then combined this with our cellular markers and clustered it together to get these select states. So this is what our select states look like on a UMAP, as well as a heat map showing the different cellular markers as well as extracellular markers across our states. Oh, I'm sorry, the resolution looks a little different on this. Okay, so let's at least now focus in on the trail pathway. All right. So trail, or TNF-related apoptosis-inducing ligand, typically hones onto death receptors and sets off a caspase cascade for cell death. That, paired with its ability to selectively kill cancer cells, got this recombinant protein into clinical trials around a decade ago. But there's a lot of resistance to it, and it wasn't very effective. So one of the main reasons for that is that it's also capable of setting off a pro-survival mechanism with activation of AKT, P38, and mTOR pathways. So we wanted to better understand how this ligand was distributed in CRC and where it was orchestrating pro-survival tumorigenic pathways using phosphorylated S6 as an mTOR readout. So we actually found trail around different cells extracellularly, perhaps priming it for signaling, but it was membrane-bound and highly correlated to phospho-S6 signal in specific tumor regions with high pan-CK and Wnt activation. So over there we see a high correlation. Again, sorry about the resolution on this. Did not look like that before. And here we see in these images a trail in cyan as well as phospho-S6 in red. Okay, and looking at single-cell RNA data, we see higher levels of trail receptor in the corner over here, expressed in poorly differentiated epithelia, which likely corresponds to our identified pan-CK high cells. All right. Switching gears, we know that proteases play a major role in signal attenuation and ECM remodeling in cancers, and we therefore assayed about 4 of them. So interestingly, we see them highly correlated in cell proximal extracellular space, especially around CRC epithelia. So in the blue box over there, again, I'm sorry for the resolution on this, we see granzyme B, HTRA1, and MMP9 come up together extracellularly across a lot of different CRC states, especially in the epithelial, around epithelial cells. So granzyme B, which is typically talked about in the cytotoxic T cell context, was found highly correlated to MMP9 extracellularly, which is a protease with roles in immune modulation, infiltration, and invasion. So let's focus in on two contexts, neutrophils and B cells, in which we see them both enriched cellularly as well as extracellularly. Right. So neutrophils, and especially tumor-associated neutrophils, or TANs, are actually known to pump out MMP9. But granzyme B positive neutrophils are not as well known outside of TB, for example, granulomas. So we're actually detecting pretty high enrichment, and certainly higher than T cell levels of granzyme B in neutrophils, alongside MMP9 as well. So we do think that these neutrophils are playing a key role in tumor progression, so we're following up and further characterizing it. Okay. Another unexpected context in which we see proteases, and specifically MMP9 is in tertiary lymphoid aggregates. So here we're looking at B and T cells, in red, CD20, and in green, CD3. And more specifically, we see it in CD40 positive B cells with high GLUT1, which is a glucose transporter. And in cyan, we can look at MMP9, which is kind of dotted all over this tertiary lymphoid aggregates. So usually MMP9 is associated with B cells in the context of B cell lymphomas, but we're seeing similar patterns in these CRC tertiary lymphoid aggregates, suggesting some sort of B cell dysregulation mechanisms happening, right? The other clue to what MMP9 might be doing in these TLAs is its role in degrading key chemoattractants, like CXCL12, for example. So if we refer back to our single-cell RNA data, even though we see in the protein level MMP9 is enriched in TLAs, it's definitely not expressed there. So we see over here CD40 expressed in B cells at the top, but MMP9 is pretty constrained to macrophages. Okay, cool. So to quickly summarize, I hope I've impressed upon you the importance of looking outside cells and the spatial patterning of key extracellular signaling proteins. And we showed you a way to capture these cell proximal extracellular patterns and signaling states, if you will, as well as we shared a couple of brief stories on TRAIL as well as proteases. And finally, a really big thank you to a lot of people, especially Dane, Vyravan, and Wilfred for their support, along with Sean Bendel, our collaborators, and Ian Tan, and of course, Sham. Thank you so much. Yeah.
Thank you. We have time for one question.
Yeah.
Yep. Thank you for the great talk. More of a technical question onto your deep learning model that you use to help with the I guess, remove the bleeding artifacts.
Yeah.
Can you explain just a bit more in detail how that works, how you
So our collaborators developed it in their lab. So essentially, it was trained on a bunch of different spatial protein data sets, spatial proteomic data sets, as well as cell type annotations. So essentially, what it has a unit architecture, and it takes every marker's image, and then we also put in our own annotations of cell types and marker positivity, and it kind of outputs a confidence score based on the segmentation masks as well as the raw images themselves. Yeah, thank you.
Okay, thank you.
Thank you.
And big applause for Matt. And the next speaker is Xiaohan Xu, please.
Hello, everybody. My name is Xiaohan. I'm a first-year PhD student in the Tuang lab at the University of Queensland. Today, I'm going to talk about my recent project, which is to construct a Pan-Paedcancer cell atlas and beyond. To begin with, I want to give a brief introduction about our center, the Ian Fraser Center for Children's Immunotherapy Research. It was established to understand how pediatric immunity develops and how it is dysregulated during cancer, autoimmunity, and other infectious disease. And our lab is part of the center focusing on computational immunology, especially on analyzing large-scale single-cell and multi-omic data to uncover immune cell dynamics in pediatric diseases. And the guy in the middle is my supervisor, Dr. Kelvin Tuong, who flew back to Australia yesterday and left me alone to present in my first international conference. Okay, so no more delays, let's get started. Pediatric cancer is one of the leading causes of children's death. If we focus on Australia, we can see that cancer is responsible for 19% of fatalities between 2015 and 2017, standing as the secondary cause of death among children. Given these worrying statistics, it is important to deepen our understanding of pediatric cancers to develop more effective treatments. However, pediatric cancer is very rare. The incidence rate is 0.017%. Moreover, children are not simply small adults and can't be treated as adults. In fact, we probably don't have enough knowledge in children too. If we look at HCA, children are currently underrepresented, and last I checked, they only constitute 60% of the entire HCA. For us to be able to treat childhood cancer better, we need to make an effort to create a more comprehensive dataset, which can act as a reference for future pediatric cancer research. And that is what I set out to do, to first look at what has already been published. So we conducted a comprehensive literature search using Google Scholar for published single-cell RNA sequencing datasets of pediatric cancers up to March 2024. A valid dataset should have generated sequencing data directly from patient samples, and the patient should be under 18 at the time of sampling. As a result, we have identified 47 single-cell data. And these 47 datasets cover 8 major pediatric cancer types. Among these, peripheral nervous system tumors were the most profiled in terms of the number of recruited patients. Notably, lymphoma, a type of hematological cancer that forms the solid tumors in lymphoid organs and is the second most common childhood cancer globally, was relatively understudied by single-cell sequencing. This is probably because surgical resection of affected lymph nodes is not the mainstay of treatment, limiting access to research samples. In contrast, central nervous system and peripheral nervous system tumors often undergo surgical resections as the first-line therapy, providing a route for accessing tissue samples for single-cell RNA sequencing. Overall, these datasets were sourced from various genomics data repositories, with the Gene Expression Omnibus, or GEO, being the most widely used. Some datasets were deposited in repositories with restricted access, for example, on the European Genome Phenome Archive, or EGA. Droplet-based 10x genomics was the most popular single-cell technology adopted by more than 47% of studies, likely because of the high throughput capabilities of the technology compared with other competing technologies at the time of the study. However, only around 60% of the published datasets could be downloaded and processed, used for downstream analysis, while I was able to generate a metadataset covering over 1.3 million cells from 488 pediatric cancer samples. My attempt to curate these datasets revealed major issues, limiting the utility of the resulting datasets. One of the biggest problems we ran into is the inconsistency in the future space across studies, largely due to the varying interpretations of processed data. Some authors deposit row count matrices that include all barcodes, or only the barcodes identified as cells, both of which are usable. However, others provide normalized or transformed matrices that cannot be reverted to the raw format, often due to the unclear pre-processing descriptions. In many cases, processed data also include only genes expressed within a certain threshold, discarding potentially valuable information. All these inconsistencies complicate the integration process and result in significant feature loss. So in the end, my Pan-Paedcancer single-cell sequencing data only kept about 7,600 genes, more than 77% less than the usual 33 to 36,000 genes we would expect, which really limits what we can learn from it. Second, there's a general lack of inclusion of healthy controls. It has been shown that when distinguish altered cell states between disease cohorts and healthy controls, it is very important to use a combination of data from publicly available healthy single-cell atlases as well as matched internal controls, namely healthy control data generated as part of the study. Lack of appropriate healthy controls may lead to an increase in the false positive findings and can misconstrue the underlying biology. Another major issue is the absence of essential cell-level or sample-level metadata. More than 72% of datasets do not provide any cell annotations for their single-cell study, limiting both the interpretability and the reproducibility of their research findings. Additionally, 70% of the datasets lacked essential clinical metadata such as age, sex, disease stage, and treatment history for the patients. While comprehensive clinical information is very crucial for patient stratification and for enabling robust comparisons across clinical subgroups. Accordingly, we propose a minimum standard pathway for depositing or uploading single-cell RNA sequencing data. As the accuracy and completeness of the human genome reference continue to improve, depositing single-cell RNA sequencing data in the format of raw sequence reads is preferred and will future-proof this data. However, due to ethical concerns and data privacy, raw sequence reads could be patient identifiable, so we usually deposit single-cell data in the format of gene expression matrices. During read alignment, cell calling algorithms are used to differentiate cell-associated barcodes and background noise. Given the ongoing improvements in cell calling algorithms, unfiltered gene expression matrices are preferred. Meanwhile, it is crucial that these matrices contain raw counts as integers rather than normalized or transformed scaled counts as floats. Moreover, files containing critical information such as patient ID, patient's clinical features, and cell annotations should be provided where possible. We hope that data standards institutes and repositories such as GEO and EGA can implement these data upload practices to ensure public available datasets are more accessible, reproducible, and valuable for future research. And now becomes the beyond part of my presentation. I'm helping to set up and analyze an Australian Pediatric Immune Cell Atlas, or PICA, which aims to conduct single-cell sequencing on 1,000 healthy pediatric samples. Because PICA focuses on healthy children, we have a companion project called Pikachu, standing for Pediatric Immune Cell Atlas for Children's Immunotherapy Innovation, working with the pediatricians as well for cancer, autoimmunity, and immunodeficiency. We hope that this can contribute to HCA in a few years' time. With that, I would like to thank all the people from our lab and everyone involved in the Pikachu project. Lastly, thank you all for your attention and time. I'm happy to take any questions.
Thank you. We have time for one question.
First, I just want to say you've done an awesome job in your first international presentation.
Thank you.
You didn't need your advisor.
Oh, I'm super nervous.
I think the challenges you faced with integrating the public pediatric cancer data are the same challenges we face in the Human Cell Atlas, and that's actually the hardest part. Integrating the data is also hard, but it actually can happen faster sometimes. One thing that helped us is, if you've got some missing barcodes, right, because some of those studies, they've already done their QC. But what we found is that the QC that people normally do is very loose QC. They throw away only the truly, complete useless cells. But they still keep a lot of cells that they should not keep. So it won't hurt you so much, I think. You should probably impose even stricter QC cutoffs on all the samples, and then throw away even more barcodes than they threw away. So then you'll have a uniformly QC'd data set. But some of the other things you mentioned, yeah, it's tough to deal with.
Yep, I totally agree with you.
Okay, big applause for you. And last but not least is Go Sato. Thank you.
Hi, hello everyone. This is Go Sato from the University of Tokyo, Japan. And I'm very happy to have the opportunity to speak to you today. I'm going to talk about our recent research on how somatic mutations affect immune cell function in non-cancer contexts. By integrating genomics and single-cell transcriptomics data, we were able to dissect the cell type-specific impacts. There are broadly two types of genetic variation in our genome, germline and somatic. Unlike germline variation, somatic variation arise after birth, accumulated in a subset of cells due to aging or environmental factors. And the somatic mutations have long been a central focus in cancer research, but in recent years, they have also been recognized for their roles in non-cancer diseases. In particular, somatic mutations in blood cells are referred to as coronary hematopoiesis. This is a common age-related phenomenon that can be observed even in healthy individuals. And coronary hematopoiesis includes several types of alterations. Among them, we focused on mosaic chromosomal alterations, MCA. MCA is a large-scale structural abnormality at the chromosome level, including copy number gain, loss, copy neutral loss of heterozygosity, and loss of the Y chromosome. These somatic mutations can be systematically screened using SNP genotyping data, and large-scale biobank studies have made it possible to identify individuals carrying somatic mutations and to investigate which types of somatic mutations are associated with specific diseases. While such population-level findings are accumulating, but the underlying biological mechanisms remain poorly understood. To address this gap, we integrated single-cell analysis with genomic approaches to gain mechanistic insights. We identified individuals carrying somatic mutations through genomic screening and performed single-cell RNA sequencing for their blood immune cells. This enabled us to distinguish somatically mutated cells from normal cells at single-cell resolution. As a result, we could characterize both the cell type-specific distribution of somatic mutations and their functional consequences within the affected immune cells. In our study, we constructed large-scale single-cell immune cell atlases by integrating genomics and single-cell transcriptomics data from 235 Japanese individuals, including 88 COVID-19 patients. Of course, this resource can be used for genomic analysis, such as eQTR analysis, but at the same time, this resource enabled single-cell deconvolution of autosomal MCAs or loss of the Y chromosome in male subjects. In our datasets, we performed genomic screening using SNP genotyping data and detected autosomal MCAs in 5 COVID-19 patients and 1 healthy individual. Among them, 2 COVID-19 patients showed strong cell type-specific enrichment of the mutated cells, making them particularly informative for understanding the biological impact of somatic mutations. For example, in 1 COVID-19 patient, we observed 2 distinct clones carrying different chromosomal alterations, and both of which were enriched in monocytes and dendritic cells. And we performed differential gene expression analysis comparing mutated monocytes and normal monocytes from the same individual. And in monocytes carrying 1P loss, genes located on 1P were consistently downregulated. This demonstrating that our single-cell deconvolution accurately identify the mutated cells. In conversely, upregulated genes in the 1P loss monocytes were significantly enriched in immune-related pathways such as cytokine production or inflammatory signaling. This provides a concrete example of how somatic mutation can alter the functional state of immune cells. Next, I'd like to introduce loss of the Y chromosome, LOY. LOY is the most common somatic mutation observed in males. In our dataset, we classified all male-derived immune cells into two groups: normal cells with detectable Y gene expression and LOY cells lacking such expression. And we found that LOY cells were enriched in monocytes and also showed a COVID-19 specific enrichment in dendritic cells, suggesting disease-dependent skewing of clonal composition. And for the detection of the mutated cells with autosomal MCAs, we look at aggregated gene expression and allelic imbalance of heterozygous SNPs within the affected genomic region. But in contrast, LOY cell detection is more simple and based on the presence or absence of Y gene expression. So this simplicity makes it easy to apply our classification approach to external datasets. So we applied it to large-scale PBMC datasets, 1K1K and IDA. And in both datasets, the enrichment of LOY cells in monocytes were consistently replicated. And furthermore, the proportion of LOY cells within each individual increased with age across all datasets. And male individuals from 1K1K showed particularly higher LOY cell fraction. Of course, we cannot completely rule out technical differences in sample preparation or data construction, but this may suggest that Europeans are more vulnerable to LOY, and ancestral diversity may affect somatic mutation patterns. Lastly, we applied our single-cell deconvolution approach to a rare autoimmune disease, neuromyelitis optica spectrum disorder, NMOSD, which affects the central nervous system. And we used a separate Japanese GBAS cohort and detected significantly increased frequency of autosomal MCAs in NMOSD cases. And among the detected MCAs, 21q loss were specifically enriched in NMOSD cases. So we performed single-cell RNA sequencing for 2 NMOSD cases carrying 21q loss, and we found that the mutated cells were significantly enriched in CD4 positive T cells. These CD4 positive T cells with 21q loss showed downregulation of type 1 interferon-related genes, including IFNA1 and IFNA2 located on chromosome 21, and other interferon-related genes located on the other chromosomes. These findings suggest that disease-specific somatic mutations in CD4 positive T cells may disrupt interferon signaling and potentially contribute to the disease pathogenesis. In summary, by integrating genomics and single-cell transcriptomics data, we achieved high-resolution dissection of somatic mutations in blood immune cells and identified precise mapping of their cell type specificity and functional impacts. And our findings also highlight the importance of developing comprehensive single-cell atlases that capture diverse ancestral diversity and multiple disease conditions. I'd like to thank all of my collaborators for their contributions, and thank you for kind attention.
We have time for questions. Any questions?
Cool. That's a really dramatic effect you saw from chromosomal aberration. So that's amazing. The kind of cell state bias is huge, right? In your UMAPs, it looks like the ones with loss of chromosome Y, or they're very, very biased cell state composition.
What's the huge?
The bias in the cell state composition.
Bias.
They seem to congregate in specific cell states.
Yep.
And specific cell types also you mentioned for different chromosomes. Is that coming from lineage bias, that the mutation happens in some progenitor cell and then there's a lineage bias in differentiation? Or do you think the chromosome loss happens after differentiation?
And in our interpretation, somatic mutation in hematopoietic stem cells in bone marrow, something, causes the more growing or many mutated cells in peripheral blood, I think.
Okay, so you think the chromosome loss happens in the HSCs?
Yes.
And then there's a dramatic lineage bias then?
Yes, I think so.
Okay. Interesting.
Okay, well, we're done. Well, let's thank all the speakers for this afternoon again. I don't know if Esther, we have any closing slides. Is she here? Okay. But if not, I think you guys saw the collection of the thanks slides from a few days ago. Okay, yesterday. Maybe, there you go. Okay, some nice slides, a bit quick. All right. No, that's... Okay, well, I see. Let's go forward. Can you show the next slide? Okay, I think this is probably the message that we also want to end. We ended with this slide yesterday. Again, it's an iteration that please join the HCA Hub. And it's a great way to connect. And I think all the discussions we had in the earlier session, again, it's a good way to see who's doing what, and we can help you to make that connection for you. And again, we would encourage you to lead and really engage with the community. And we also want to hear from you. And if you didn't have a chance to take the survey, please take a QR code. Appreciate your feedback, how we can improve. Also have been sent out, so please check and fill out your survey. Thanks again. Sham, do you want to say anything?
Not much more to add. Thank you so much to everyone for bringing so much energy and so many ideas and great presentations to the meeting. It's great to see new faces and also some of the regulars in HCA. I really, really hope that this won't be just scientific talks. It'll also be ways to meet each other and form collaborations. And I hope that even as people walk out of here, there will be some conversations about new projects that can be started. And that's really the goal of this kind of meeting. Thank you all once again.
Thank you.